Hi everyone, this is lecture 2 of topic 7 of the human anatomy class. We have started learning about the nervous system. There are two types of cells included in the neuroglia in the peripheral nervous system. The first one is called satellite cells. The function of the satellite cells is to surround the neurons and they end up protecting the cell bodies or soma in the PNS. Schwann cells are the second type of neuroglial cells present in the PNS. These are also known as neurolemmocytes, and their main function is to produce myelin sheath around the neuronal axons in the PNS. So the oligodendrocytes produce the myelin sheath in the CNS, and the Schwann cells produce the myelin sheath in the PNS. This figure shows the satellite cells surrounding the soma and protecting it, while the Schwann cells in the axon region making the myelin sheath. This is a summary of the neuroglia as found in the CNS and the PNS. The CNS includes 4 types of cells and the PNS includes 2 cells. So total of 6 types of cells in the neuroglia. Moving to the neurons or the nerve cells. The nerve cells are the longest cells in the human body, and their most important characteristic is the ability to transmit messages or electrical signals, and these electrical signals are known as nerve impulses or neuronal action potential or simply action potential. If you look at a neuron, there are two main regions in the body of the neuron. The first one is called the cell body or the soma or the cyton. This is the main metabolic center of the cell, contains nucleus and other organelles. The other region of the neuron includes processes. Processes are fibers which extend from the cell body, and these include the axon and the dendrites. The soma or the cell body of the neuron contains some of the following organelles. The first one are Nissl bodies, which are specialized rough endoplasmic reticulum with ribosomes, so they are mainly involved in protein synthesis. The second one are neurofibrils, which constitute the cytoskeleton, so they help the neuron in maintaining its shape. And of course, the nucleus, which controls all of the neuronal functions, and the nucleus has a large nucleolus. This is a diagrammatic representation of a nerve cell or a neuron. This is a micrograph of a neuron with the cell body and the dendrites visible. Neurons have two types of processes outside the cell body. The first one is dendrites. These are branched processes and they are responsible for conducting the impulses or the action potential towards the cell body, so always in the direction of the cell body. These are short and multiple. Axon is a single process that conducts the action potential away from the cell body. Neurons have only one axon and it comes out from the cell body at a conical junction point known as the axon hillock. The axon of the neuron comes out of the cell body at the triangular junction called the axon hillock. The axon hillock is the initiation center. It is the one that starts the chemical impulse transmission. The action potential travels the length of the axon, and then the axon ends in enlarged club-shaped endings which are called axon terminals, and the axon terminals are located at the ends of small terminal branches called the telodendria. There are special synaptic vesicles that are present in the axon terminals, and these contain chemicals called neurotransmitters such as acetylcholine, which do the chemical talking between nerves and from one neuron and another neuron, or from the nerve to the muscle. So notice the location of the axon hillock, the axon terminal, and the telodendria. The axons are covered by myelin sheath. The myelin sheath is produced by Schwann cells in the PNS and oligodendrocytes in the CNS. The myelin sheath is a whitish fatty material. It is actually an extension of the cell membrane and it covers the axons in a spiral manner. So there are gaps in the myelin sheath along the length of the axon. The function of myelin sheath is first to insulate the neurons and second to increase the speed of the action potential conduction, because the electrical signal jumps from one node of Ranvier to the next. It doesn't have to travel through the entire length, but it can just hop. So this figure shows the Schwann cell, the myelin sheath which is formed by the Schwann cell. Look at the gaps along the length of the axon where the myelin sheath is absent. That is known as the node of Ranvier, and the electrical signals simply hop from one node to the next one. Neurons are classified into three types based on their structure. First one is multipolar neuron. Multi means many. So there are many extensions coming out from the cell body. There is one axon and then multiple dendrites. This is the most common type of neuron, and all motor and interneurons are multipolar. The second one is a bipolar neuron. Bi means two. So in this neuron there is one axon and one dendrite only, and it is present only in special sense organs such as nose or the eye. The third one is a pseudo-unipolar neuron. Uni means one, but pseudo means fake. So these neurons, they have a short single fake process leaving the cell body. We call it fake because the process, when it comes out, it looks like one, but then it immediately branches out. So that's why the fake or pretending to be one process neuron. And this is found in sensory neurons found in the PNS ganglia. This figure illustrates the multipolar, bipolar, and pseudo-unipolar neurons. Functionally, neurons are classified into three categories. The first one is a sensory or the afferent neuron. These neurons carry impulses to the CNS, that means always in the direction of CNS from the sensory receptors. The second one is motor or efferent neurons, which carry impulses away from the CNS towards the body. The third one is the interneuron or the association neurons, which are translators between the sensory and the motor. So these are found in neural pathways in the CNS, and they connect the sensory and motor neurons. In this figure, the sensory neuron is shown in turquoise blue, the motor neuron in red, and the interneuron in green. So the most important feature of nervous tissue, which sets it apart from other tissues, is its ability to generate and transmit electrical signals. And this is possible because the nervous tissue is irritable. That means it can respond to stimulus, and it can also conduct, so it can transmit the electrical signal. This slide includes a list of terms and their definitions that all of you need to memorize before we move to the next lecture. The first term is nuclei. Do not get confused with this nuclei and the nucleus inside a cell. This nuclei means a collection of the cell bodies or soma of the neuron in the CNS. While the same thing in the PNS, that means a collection of the soma of neuron in the PNS, it's not known as nuclei, it is known as ganglia. Tracts versus nerves. Tracts are nerve fibers or axons in the CNS, while nerves are axons in the PNS. Gray matter includes the cell bodies, that means the soma, and unmyelinated fibers, that means axons which are not covered by myelin sheath. While white matter includes only the myelinated axons, that means axons that are covered by myelin sheath. This slide shows the gray matter and the white matter in sections of spinal cord and brain. A synapse means the junction point of a neuron and a target cell. It could be two neurons, or it could be a neuron and its effector organ. Usually a synapse consists of three parts. The first one is known as the presynaptic neuron. This neuron is sending messages. The second one is a postsynaptic neuron or an effector organ. This is the one that is receiving the message. And the third component is synaptic cleft. This is the gap between the adjacent neurons, and this is usually filled with interstitial fluid called synaptic fluid. There are two types of synapses, or two ways in which synaptic communication occurs. The first one is called a vesicular synapse or a chemical synapse. This synapse uses vesicles, synaptic vesicles, and there is neurotransmitter such as acetylcholine inside the synaptic vesicles. The second type of synapse does not include the vesicles, so it is known as a non-vesicular synapse. It is an electrical synapse and it involves the flow of ions. The figure illustrates the two principal kinds of synapses, electrical and chemical. Vesicular synapse involves the use of neurotransmitters in the synaptic vesicles. The neurotransmitter diffuses across the synaptic cleft and binds to receptors on the postsynaptic membrane, and this causes changes in the permeability of the postsynaptic membrane, and the postsynaptic neuron becomes more permeable to sodium, so there are ions that move in and out, and this results in an action potential in the next neuron. This figure shows how one presynaptic neuron could be communicating with multiple postsynaptic neurons. In non-vesicular synapse, the electrical signal can be conveyed in any direction, unlike the vesicular synapse where the electrical signal could be conveyed only in one direction. And here, the presynaptic membrane is very tightly bound to the membrane of the postsynaptic neuron. In the vesicular synapse, these two membranes do not touch. And because there is binding here, it permits the movement of ions from one neuron to the next.